This is the second podcast on the hydrogen bonding that exists between DNA strands. And we're just talking about water from part one. And we were talking about the fact that the electrons in the arrangement between the bonding between oxygen and hydrogens are pulled much more closely to the oxygen. This is going to lead to an important feature, and that is that the oxygen is going to be ever so slightly negative because it has electrons pulled towards it, and the hydrogens are going to be ever so slightly positive because they've had their electrons withdrawn away from them. Electrons, of course, being negative. So oxygen is ever so slightly negative, which we write as delta negative, and hydrogen is ever so slightly positive, which we write as delta positive. This is the basis of the hydrogen bond, because if you have two separate charges, as we've mentioned, plus attracts minus, and so here is the hydrogen bond link between water molecules, where the H plus, which is not actually a charged particle, but a momentary positive charge as the electrons are moving around, is attracted to the momentary negative charge of an oxygen molecule, a water molecule next door, and vice versa. So oxygen here would be attracted, for example, to an H plus of water over here, and this H plus is going to be attracted to an oxygen over here. And you can see that water, in fact, due to hydrogen bonding, creates this mosaic of hydrogen bonding between billions and trillions of water molecules, where this slightly positive hydrogen bond to a slightly negative oxygen. Last thing to say about hydrogen bonding is in water it's a very fast transient bond. Because water's moving around, the hydrogen bonds happen and then go, happen and then go, happen and then go. But because there's so many trillions of water molecules, the truth is that at any one time they are all bound to each other in some form or another. If we could take a photo and get them all to stop, there would be trillions and trillions of water molecules bound together by hydrogen bonds. And if you let the time run on for a millisecond and then took a photo, there would still be trillions of hydrogen bonds, but they'd be slightly different between different water molecules. And hydrogen bonds in water are constantly changing, but always there's enough there to keep cohesion. And so hydrogen bonding also is responsible for keeping the strands of DNA together. And the main difference here is that the strands are not moving like water, and so the hydrogen bonds are more fixed, and that actually happens in ice. When water freezes, hydrogen bonds fix and it creates a rigid solid structure. I want us to look now then at a DNA strand and see if we can identify the two strands from a model of the DNA molecule, which goes into a little more detail than we've seen before, and perhaps to allay your fears that it's not as complex as it might seem. We have, first of all, the cast of characters that we are aware of from our study so far: hydrogen, oxygen, nitrogen, carbon and phosphorus. Hopefully you should know that the yellows here and here and here are going to be the phosphate groups, and the nitrogens, the blues, are going to be part of nitrogenous bases, okay, which you can see focus more on the inside of the molecule. And with that in mind, you can see if we were to follow these yellows, we actually can make out the three-dimensional strand being turned into the two-dimensional alpha helix that we know so well. Get rid of those. And that, in fact, is a very much easier shape to follow. But can you see we've just turned all that complexity into the alpha helix that we know so well. And as a result of that, and the fact that we can see the nitrogenous bases, we can see the base pairing. And therefore, we can glean that the hydrogen bonds must be between them, like that. And in fact, the diagram here helpfully shows us these hydrogen bonds between the T and the A's and the G's and the C's, and you can see that there's 2 for A and T and 3 for C and G. But what I want us to focus on, bearing in mind what we've learnt about water and the fact that it's the electronegativity of oxygen pulling the electrons towards itself that creates what we call a dipole, which is just like on the Earth, North and South Pole, we have 2 different charges. So instead of north and south, it's plus and minus. And I want to focus on the fact that there are 3 main players in the hydrogen bonding in DNA. We've already met hydrogen and oxygen in water, and it's going to be very similar, but we also bring nitrogen into play. And it is the collaboration of hydrogen bonding between these 3 atoms in the DNA that cause the hydrogen bonding. And this can be best seen if we look at a detailed version of our purine binding to a pyrimidine. And we can see here that this must be guanine to cytosine, because there is 1, 2, 3 hydrogen bonds. So how does this happen? Well, it's very similar to water. Here we have oxygen, which is going to be ever so slightly negative because it is pulling electrons towards itself from the carbon. And hydrogen is going to be ever so slightly positive because nitrogen is pulling electrons from it. And therefore we have plus and minus. And because the purines and the pyrimidines fit each other like a jigsaw, they're able to get very close to each other, and that allows the hydrogen bond to form. And here it is. Likewise, here we have a hydrogen bond between hydrogen and nitrogen. Hydrogen is going to be delta positive, nitrogen is going to be delta negative, and then there's the hydrogen bond. And lastly, same again, we have hydrogen, which is going to be delta positive, and oxygen, which is going to be delta negative, and then another hydrogen bond. And so, in just the same way as water, we have a differentiation of charge, slightly positive, slightly negative, which creates an attraction, and that attraction is enough to hold the DNA strands together. I mentioned it was a bit like a jigsaw puzzle coming together, and this is undoubtedly why we have the arrangement of the antiparallel strands as well. And if we can just keep these both in shot, can we see here the pattern of the oxygen here at the top for the purine, and the oxygen here at the bottom. And we have this inverted flip, if you like. So this one is the right way up, and this one is flipped, so that the oxygen is gone to the bottom. And it's that arrangement which creates the 3 hydrogen bonds which allow bonding to take place. And so this here, if I was going to be accurate to this diagram, I need to add a hydrogen bond, is the bonding of guanine to cytosine. And here we would have an oxygen at the top, and here we'd have an oxygen at the bottom, and we'd have nitrogens, and the hydrogens, and they therefore form the atomic arrangement which leads to the hydrogen bonds. And so hydrogen bonding is very important in the bonding of DNA strands because it creates, due to the electronegativities of the atoms involved, slight dipoles, positive and negative areas of charge, which are able to fit together for each of the purines and the pyrimidines in a jigsaw type fashion. Imagine that was a jigsaw, and we know that would fit in to that. And so the purines and the pyrimidines are able to fit in with each other due to the fact that hydrogen bonds can form between the oxygens and hydrogens or the nitrogens and hydrogens. And the only way they can do that is if one nucleotide is the right way up and the other is flipped, with the oxygens at each end forming the electronegativity of the positive charge. So that is an overall look of the hydrogen bonding that exists between the DNA strands. What do I want you to take away from this? Well, first of all, to understand the hydrogen bond which is formed between two atoms which have a difference in charge due to electronegativity differences. And secondly, that the purine and the pyrimidine they form an interlocking hydrogen bond arrangement because of their shapes and where the oxygens and the nitrogens are. And because of that, the way that they would fit together would be for one of the nucleotides to be the right way up as we draw it, the other one to be inverted, and that explains why we have the 5' to 3' and 3' to 5' in this diagram, one strand going one way and the other going the other.